Just to do a quick review, let me turn off the lights. We're talking about the fact that we've got to be able, anytime, so remember, to shuttle nitrogen within the same cell, what amino acid is the shuttle for nitrogen? If it's inside the same cell. That's really important. If it's inside the same cell versus if it's going to have to go into the bloodstream. What amino acid is it that does intracellular nitrogen trafficking? It's what? It uses aminotransferases. Which amino acid is a substrate for every single aminotransferase? Glutamate. Glutamate is one way to shuttle nitrogen inside the same cell. But there are two different amino acids that anytime you had to move a nitrogen from one part of the body to a different part of the body via the bloodstream. Well, no, this is we're talking about the amino acids. Those two amino acids used to shuttle them, and that's one of the reasons why they're two highest in the bloodstream, what are those two amino acids? If it's going from one cell to another cell via the bloodstream. Alanine, because that's for your muscles. And all the other tissues use... Nope, not glutamate. So glutamate was for intracellular. Glutamine. Remember? So this is what this is showing. I mean, in fact, it's right, by the way, people, it's the first sentence. Not even a sentence. Okay. It's a long week and it's only Monday, right? So, but no, it's glutamine. Okay, glutamine and alanine are used for intercellular nitrogen trafficking. Intracellular is glutamate because you can just use an aminotransferase. Okay. However, the way that this is set up, it's a little different, in the sense that we have. This would be like the first tissue. Let's say, for example, maybe your brain your brain, we'll say. And it's got to get rid of a nitrogen. The way that it does it, once again, is we have glutamate at the expense of an ATP, so it's a kinase. It's a synthetase. It's going to go, and it's a two-step process. So that's why it's a synthetase, not just a kinase. Okay? But it's the same enzyme. They've got listed here twice. But it's going to turn a glutamate using up an ATP and an ammonia, because that's the nitrogen that you're trying to get rid of, to make glutamine. Glutamine now leaves the brain in this instance, goes into the blood, where it can go to the liver, comes back into the liver, and it was glutaminase that takes off that. And we already talked about the reason why it couldn't be an aminotransferase, because if it was an aminotransferase, we would just be making other glutamates, we wouldn't get rid of it. But it's glutaminase. Plus, this aminotransferase is typically we're working at this nitrogen, and here we're working at getting rid of what would be the amide nitrogen. It's called the amidino group. Okay, so this nitrogen is the one that gets rid of, it takes the water. You can imagine the oxygen from the water comes and smacks that carbon, boom, side effects optional. Of course, then, we had a tetraethyl intermediate and we break it down and we kick off an H4. But then H4 inside the liver cell, the hepatocyte's going to go to the urea cycle, okay? And we have glutamate, and of course, then glutamate can be utilized for lots of different things. But glutamine is non-toxic. There's a reason why glutamine and alanine are much higher than other amino acids in the blood. That's because it's used for nitrogen shuttling. And glutamine is also a precursor for things later on that we'll talk about, for some of the other biosynthetic things. So that's why it's important. So glutamine synthetase is an important enzyme, as is glutaminase, and glutaminase is in the liver.
So this is just kind of showing going from glutamate to glutamine back to glutamate?
Right, because, and what's not shown here is, up to this point, this is in some tissue other than skeletal muscle. Skeletal muscle uses alanine. And so the whole goal is, for example, your brain trying to get rid of a nitrogen, because you do not want nitrogen to stick around. And we'll talk about the reason why in a moment. And so the way it does that is we take a glutamate from the brain, we add on the nitrogen it's trying to get rid of. Now it's glutamine. Glutamine is non-toxic. Glutamine now goes out, goes into the blood, gets to the liver, where then the reverse is done, but the reverse reaction is done by glutaminase. Okay, so now the nitrogen can go into the urea cycle, and then glutamate itself can be used for a whole host of things.
And glutamine gets turned into glutamate only in the liver?
Right, in this instance. Mm-hmm. For this purpose, yes. if the purpose is getting rid of nitrogen. All right, now the muscles use alanine, and we just barely started to talk about this because we were out of time. And remember, it used alanine because of the fact that the muscles under anaerobic conditions convert convert lactic acid to pyruvate and pyruvate to alanine. Okay, so there's lots of alanine around, but it's the same principle here. Okay, and so that's why this is called the glucose alanine cycle, because it's done in concert with the Cori cycle, which was lactate and glucose. Remember that was the whole idea, that your muscles could then utilize lactate for energy whenever sugar is depleted under anaerobic conditions. Okay, so this is what it's all trying to show. And now the enzyme, please note, so here we have pyruvate to alanine using alanine aminotransferase. See, every aminotransferase has a glutamate that's going to be alpha-ketoglutarate, or, well, see, down here, alpha-ketoglutarate becoming glutamate. The alanine is non-toxic as well, so it goes into the blood, goes to the hepatocyte, where the hepatocyte uses alanine aminotransferase. It's the same enzyme, it's just a different tissue. It's going to be the hepatic alanine aminotransferase. And so that's the reason why we have alanine hooking up with alpha-ketoglutarate to make pyruvate and glutamate. Of course, glutamate then can get rid of the nitrogen for the urea cycle. Pyruvate can go through gluconeogenesis to make glucose, which can go and replenish the supply that the skeletal muscles need. Okay, and actually I've been saying all this, but this is just in case I was to overlook something, to remind myself as well. So first, that's very important. Why do we want something non-toxic in our blood? Sometimes people overlook the obvious. We use alanine, it's non-toxic, so therefore we don't die. Okay, alanine and glutamine. Your muscles are working out, you're feeling the burn, so there's not enough oxygen around, there's not enough sugar around, we're going under anaerobic conditions, so we're going to make lots and lots of pyruvate and lactate. Okay, so you can completely circumvent the citric acid cycle. This pyruvate builds up, we can just use the alanine aminotransferase to make it into alanine, boom, which then can go on to the liver, and the reverse happens with alanine aminotransferase in the liver, and so therefore the liver can then go through gluconeogenesis from the pyruvate that it got, and so it's replenishing the muscle's sugar supply. It's quite efficient.
So, would it be, I don't know how to ask the question, but what about milk? What about actually getting lactose from milk?
Lactose is different than lactic acid.
Oh.
Right? Because lactose is a sugar, lactic acid is a carboxylic acid. Mm-hmm. But you can get energy from milk because the lactose can be broken down into sugar components using lactase, which then can undergo glycolysis and everything like that. I know, those names all start to sound really familiar. Really a lot alike. All right. Right, so by using alanine here and that Cori cycle for the lactate, we can make gluconeogenesis within the liver. So that way the liver produces ATP. The liver produces the glucose, which can then go ahead and be used for ATP production within the muscles. Okay. This is one of the reasons why we've got to get rid of ammonia. It's much more complicated than what is the fact that you don't want nitrogen around. You don't want nitrogen to overstay its welcome. Whenever it becomes fully reduced, which that's what NH3 or NH4 plus is, it's a fully reduced nitrogen versus nitrate or nitrite, which are oxidized. it's going to form ammonia. Of course, ammonia is a base, so first of all, it can affect pH. But since for example, your brain or other tissues got to get rid of this nitrogen, then it's going to start making lots and lots of glutamine, right? Because it doesn't want to mess up the pH of that tissue, so it's going to make lots of glutamine. Then the problem is that they've noticed that this happens a lot with respect to coma, and it causes edema. What is edema? Swelling. And so you can imagine its use it's called osmotic pressure, and I don't know that you got to do it a lot. I mean, this is a chemical formula in which I wouldn't ask you at that point in time for osmotic pressure, but it's related to P inert, the ideal gas law. Okay, because this is a pressure. But the whole idea is, as you can imagine, if this is the cell, what is osmosis?
Water.
It's the flow of water. And so what happens, and I'm going to remember, what's the one letter code for glutamine? Q. Okay, so to get rid of that nitrogen, because you don't want to mess up the pH, you start making lots and lots of Qs. You make Qs more than you can transport them. Then what happens is the concentration of water is too low inside that brain cell. And so water is then going to come rushing in to try to level it out. So the brain cells going to become swollen, can become huge. And of course, inside for brain edema, you're really limited. Our skulls, for protection, most of us are hard-headed. And so we're limited. And so then bad things, obviously happen. So that's one of the mechanisms that they believe, that they've hypothesized is the reason why it's happening, and that you commonly see this towards this type of edema of the brain.
So in the brain, if glutamine is so toxic, in what way is it stored there?
Well, we don't really store large quantities of it. So you're going to get rid of it. Normally, in normal conditions, you would send it out to the bloodstream to go back to the liver. Okay, to go ahead and get rid of the nitrogen and stuff. But if you can't, if your body starts to break that system down, then the glutamine will build up. It's got to go someplace and build up in these tissues and these cells, and then so that it's going to swell. Another hypothesis is the fact that glutamine synthetase uses glutamate. And of course, as we already know, hopefully you've started to put two and two together, glutamate disrupts the citric acid cycle because of alpha-ketoglutarate, right? That's its keto acid. If you can imagine, which I can't draw down on here, but if you have lots of glutamine, it's taking away from the pool of glutamate you have. You're going to have to make more glutamate. You make the glutamate from alpha-ketoglutarate. You start taking away alpha-ketoglutarate, you're not going to have as much energy for the citric acid cycle. So you're going to start depleting your resources there as well, which would be bad. Okay. And then we haven't covered neurotransmitters yet, but there are some glutamates also a neurotransmitter. That's chapter 23, I think. Okay. Are there any questions? That's the end of part 1 of chapter 18.